Yeah. Well, it's very nice to be able to speak to you, even if it is by video. So, what I'd like to do is talk to you about work we've done on creating new science and engineering, and then moving it to the world. And what we've tried to do that are a couple things. One, we filed patents because that provides incentives for companies to put money into it, and it takes a lot of money to develop a lot of these things. And secondly, in many cases, we formed companies. So I'd like to go through this, and I'll go through a number of examples, but if we could just start with the first slide, and I'll talk about some general themes. So whenever we've done this, we've developed what I'll call as a platform technology. And a platform technology is something that you can use for more than one thing. In other words, I'll pick an example. I'm going to talk about drug delivery systems, but it could be other things. And drug delivery systems, basically, you could put drug A in, drug B, drug C, and in it, and so that's something that could be used for an awful lot of things. So you could license it to one company. Problem is, then you're probably not going to get the maximum utility of it because they'd only use it for their drugs. So if you had a small company, then maybe you could use, work with, say, Merck on their drugs, Pfizer on their drugs, and so forth. Anyhow, then we also want to make products. In a lot of these cases, we've published our work in really top journals like Science and Nature, and we've tied those papers to really powerful patents, seminal patents, and ideally patents that block others, which may not sound nice. But the thing is, if you want to get the kind of investment you need, usually in the medical area, you need millions, if not billions of dollars. And you won't get that from companies or investors if they think somebody else can do it. So patents end up being really important. Also, you need, particularly in medicine, which is a lot of what I'll go over, not all, but you usually want to have some type of in vivo proof of principle, some proof that at least in an animal model that it works. And finally, one of the reasons I've done this is not only do I want to get stuff out to the world, but so do my students. They've often spent 4, 5, 6 years working on their thesis or postdoctoral project, and they've made a lot of progress, and they want to see what they did really make a difference. Now, we're publishing these things, but really to get them used, you need champions. And there's no greater champion I've found than the people who actually did it themselves, because when they were a student, because they really have a great belief in it. And that's very different than just, say, licensing it to somebody sort of like a handoff and hoping that they'll do a good job. So the passion, I think, of the people in our lab who've worked on these things, that's just made all the difference in the world. So if we could go to the next slide, and I'll start on some of what we've done. So the way I got started was we were trying to develop controlled release systems, polymer systems to deliver large molecules. And the reason I was doing it was really basic research. It was because we were trying to isolate substances that were large molecules that could stop blood vessels from growing. These are called angiogenesis inhibitors. But whether it was those or really any large molecule or even any ionic molecule or most ionic molecules, they often had very short lifetimes in the body. So you'd end up needing to have a way to deliver these molecules in an unaltered form and protect them from harm. But when we started this work, pretty much everybody thought you couldn't do it. Dr. Folkman, who was my boss at the time, he asked different people, could they help us deliver these large molecules through polymers? And what everybody said is shown on the following slide. They said, you can't do it because these molecules are just too big to diffuse through the polymers. They said it's kind of like going through a wall. And so they didn't think it was possible. Now, you could make big holes in it, but then it would be through in a second, and that's not good either. Actually, the literature said the same thing, and this is shown in the next slide. That the use of polymer, this is right, a direct quote from the literature, that the use of polymer matrices for slow release systems has been virtually restricted to small molecules. And I think the only thing that I had going for me is I hadn't read any of this stuff. So I tried anyhow. And I spent several years in the lab working on this. And actually, I found about 200 different ways to get it to not work. But finally, next slide, I figured out a way to make it work. I came up with a way to make little microspheres. You can see a picture of one and cut in half. And we published in the British journal Nature on the next slide in 1976 that you could release molecules of any size for over 100 days. And then we even showed, using some engineering techniques, that you could get constant release. If we go to the next slide, you'll see that. But when I first did this, everybody told me that, and I first presented this, everybody told me that this was impossible, just like that slide I showed you earlier. And I actually received a lot of ridicule. In fact, I still remember in 1976, when I started, it was the first big talk I ever gave. And I was, I think, 27 years old. I was giving this talk in Michigan to a lot of very distinguished older chemists and engineers. And when I finished, I was very, very nervous. When I finished, I hoped people would come up and be nice to me. But actually, a whole bunch of them came up to me and they said, "We don't believe anything you just said." What they said is shown in the next slide, that I must be very naive because, again, large molecules can't diffuse through the polymers. And I was using organic solvents, which they said would destroy whatever we put in. In fact, things kind of went downhill from there, if that was possible. I was a postdoc and I tried to get research grants, and I was a chemical engineer. But the work I was doing was on cancer and on this kind of work, drug delivery. So I wrote these grants, and actually my first 9 were rejected. Very, very badly rejected, actually. They kept saying I didn't know what I was, how could an engineer like me do biological work? So that wasn't so good. And then, I was applying for faculty positions. I applied for faculty positions, and no chemical engineering department, which was my background, in the world actually was willing to offer me a faculty position. They again said that all this biology stuff that I was doing made no sense for a chemical engineer to do. I should add that today I take great satisfaction in the fact that must be half the chemical engineering departments in the world have bio in their name. But at that time, when I started doing this work, that was really, looked down upon. And then, so the only place I could get a job, actually, it turned out, was the nutrition department. And I think I got that because my boss knew the head of that department, Nevin Scrimshaw. Nevin Scrimshaw was a very famous nutritionist, but he was the kind of person who was like what I'd call almost a benevolent dictator. So he liked me, he offered me a job, but he didn't bother to ask any of the rest of the department what they thought. And that probably still would have been okay, except for one thing, that the year after I joined the department, he left. And so a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, on the next slide, this is from Michael Marletta. Mike is a very famous professor at Berkeley. He was chair of the chemistry department there for a while. He was also head of Scripps. And he was a good friend then, still a very close friend. And what Mike was, a number of years ago, I won this award, and they asked him to give a speech, and he started recollecting what it was like to be with me in 1980 at MIT. And I'll just read you one of his quotes. He said, well, one evening he went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for polymeric drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said he thought he was in a Fellini movie. So that wasn't so good either, but I'm still here at MIT, so things worked out okay, but it wasn't easy. At any rate, we did a lot of basic work. I'm not gonna go into it too much, but basically what we've discovered by using a lot of microscopy was how this worked. And the next slide shows a picture of one of these polymer systems that we developed. This is a very thin cross section. And you see it looks like a sheet. There are knife marks that you can see when we cut it with what's called a cryomicrotome. And it turns out that molecules 300 molecular weight or greater and ionic molecules cannot go from one side to the other. But if you put a drug in, it turns out that we get a phase separation and a very complex network of pores are formed. And this is shown a little bit in the next slide. And these pores, this is just one section, but we did scanning electron microscopy and serial sectioning and image analysis. And what we discovered was that these pores are interconnected, and they're also incredibly winding and tortuous. So it takes a really, really long time for the molecules to get through. What I sometimes say is it's kind of like driving a car through Boston. Boston and these pores have systems have a very high tortuosity. Tortuosity is a term that chemical engineers use to describe the windingness of porous paths. And actually, over the years, many of our students, like Elazar Edelman and Larry Brown, Ron Siegel, Mark Saltzman, they developed mathematical models at MIT to predict the tortuosity of these systems. And by so doing, it enabled us and others to design systems that could deliver drugs anywhere from a day to many, many years. And that allowed us to actually deliver these angiogenesis inhibitors, and we did isolate them. It also allowed us, on the next slide, just to deliver molecules like insulin for actually over 100 days from a single little implant. And one of the things that Dr. Folkman, who was my boss, as I mentioned, at Children's Hospital, said to me, he said, Bob, we should file for a patent. You know, that might seem commonplace today to file for a patent, but in the 1970s in Boston Children's Hospital, which is one of the best children's hospitals, maybe the best children's hospital in the world, it was very uncommon. And in fact, they never had a patent. This was the first time they ever filed for a patent. And what happened was 5 years in a row, the patent examiner rejected it. And so then the head of the licensing office at Children's Hospital came to see me and said, Bob, he said, just give up. He said, this is never going to get allowed. He said, this guy doesn't understand science, and just give up because you're costing the hospital a lot of money. But I don't like to give up. And I started thinking, how could we convince the patent examiner to allow this patent, legally, of course, and science wasn't working. But as I said in the beginning, when we first started doing this, everybody said it was impossible. It could never work. And I wondered if anybody ever wrote that down. So I did what's called a science citation search, meaning I did this in the early 1980s. And the way you do it is I look at our Nature paper in 1976, look back at it, and I saw who cited it. I'm doing this in 81 or 82. And I found a lot of papers cited it. It's a very widely cited article, but one in particular was a review article that was very, very useful, and which I thought would be very useful for the patent examiner. And if we go to the next slide, I'll just show you a quote from that. So it turns out these are 5 of the top polymer scientists in the world. And they'd written this in 1979. I had really no idea that they had written it. But I'll just read it to you. It says, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, that's a really good word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed this to the head of the technology transfer office at Children's Hospital, and he flew down and showed it to the examiner, and the patent examiner said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote that. So I wrote each of them, and they were all nice enough to write me back that they really wrote it. And we got a very broad patent, which is shown on the next slide. And with that patent, we would teach different people how to use it, license it to companies. We started companies. One of those companies, actually, just to go to the next slide, was a little company called Enzotech, which then merged to form Alkermes. The first 4 employees of this company were my former students who worked on this and were very passionate about it. And today they have like 25 products that are FDA approved or in clinical trials. It's led to very, very widely used treatments, all based on these microspheres that I showed you, for schizophrenia, for alcoholism. It's one of the most widely used treatments for opioid addiction, also for type 2 diabetes. They've got about 2,000 employees, and it's a multi-billion-dollar market cap company. So I thought I'd show you a few other examples. And next slide is another one. And this was a very nice story where David Edwards, who was at MIT, came to see me one day and asked if he could do postdoctoral work in my lab. And he is an interesting person. He mostly did mathematical modeling his whole life. And one of the things he did is he did mathematical modeling of the lung. And I had been thinking for a while that lung delivery is something that maybe we could make an impact on. By the way, lung delivery was at this time incredibly inefficient and expensive. You were lucky if you're able to aerosolize a drug and get even 2% into the lung. And you might ask, well, why? Well, everybody always thought that if you aerosolize something, it's got to be really tiny, like 2 micron diameter or less. If it was bigger, it would settle in the back of the throat, so it wouldn't get into the lung. So everybody made very, very tiny aerosols, less than 2 micron diameter. Problem is that when you aerosolize them, it's like wet sand. The surface area is so much, so they would stick together in the device, the inhaler. They'd stick together in the air, in the mouth. So by the time you get to the deep lung, you really don't have hardly any. So how did people try to solve it? Well, for many years, people tried to solve it by what I'll call mechanical engineering approaches. They would design better and better inhalers to break apart these sand-like aggregates. And they might go from 2% to 3%. Now, the thing is, that's good. I mean, it would certainly save costs because that's still 50% less, but you're still wasting 97%. And also, you could make the inhaler smaller. That's actually been one of the big problems that for so many drugs, since it was so inefficient, you'd have to carry around a giant inhaler. And of course, people won't do that. So rather than make the 50th inhaler, which people were doing, David Edwards took a very, very different approach. He basically started doing modeling and said, nobody has ever changed the aerosol design. They've always changed the inhaler design. So what we started thinking about was, well, everybody before us always made what's called a non-porous aerosol. Like that's like a meter dose inhaler or nebulizer. You're, basically nebulizing or putting water in, but there's a drug in it. They're soluble in water. But a few people had made dry powder inhalers. But the thing is that whether it was a dry powder or aqueous system, the density was always about 1 gram per cc. And what David and I started talking about was, well, what if rather than do that using a non-porous aerosol, what if we made the aerosols big but incredibly porous? And David calculated and modeled this. And the idea was that if you did that, they would because their would float different, they would still go, even though they were bigger, go to the deep lung. But because they were bigger, they don't stick together like the small ones, right? Like wet basketballs don't stick together like wet sand. Also, since they were bigger, we thought they might last longer because, the reason that aerosols usually don't last long is that there are cells called macrophages, and they eat foreign particles. Normally, they're eating like a small particle. It's like eating a small meal. It doesn't take long. But now they're basically having to look at a big particle, and that to the cell will make it take longer. So these actually not only a lot more gets in theoretically, but they also should last a lot longer. So this was theory. What happens in practice? So we had people in the lab like Justin Haynes and Noah Lotan, who was on sabbatical, and Giovanni Caponetti and Jeff Herkash, and they made some of these. And I'll just give you a sports analogy. When you look at the next slide, which is a scanning electron microscope of these aerosols, what ours look like, I mean, is maybe a way to think about it is before we started, all the aerosols people made looked like little golf balls or little baseballs. Ours looked like wiffle balls, big wiffle balls like this. But because we changed the geometry, that changed everything. And the next slide is an in vitro experiment. It basically shows that, and maybe we'll look at the right-hand part, that you get 10 times more in this Anderson impactor, 10 times more drug or aerosol deposited if they're porous compared to non-porous. And that also turns out, as we go to the next slide, to be true if you do it in an animal model, and you get 10 times more in. And also, they last longer. If you even do a human experiment, this is shown in the next slide, it can last for up to 3 days. So once again, we filed a patent, and we'll go to the next slide. And it's a very, very broad patent. It as particles, like I say, have to be light, they have to be bigger, but it covered any drug you'd ever put in it. And we started a company, again, because we wanted to get this out to the public. And that company was, we called AIR, and the next slide describes some of it. We did our initial studies in 1996. We published this in Science in 1997, started AIR in 1998. And that first year, 3 big companies gave grants to AIR to move their drugs forward. And, you know, we were in 2 clinical trials. And David didn't want to do this forever. In fact, he's got a professorship at Harvard, and he does other things too. If anybody ever went to Cafe Art Science, that's a restaurant he started. Anyhow, basically, it was we ended up selling the company, and the investors made what's called a greater than 1,000% IRR. I think it's maybe the highest in history. He also was able to use this for the developing world. You know, a lot of people sometimes say that patents are bad, but the thing is, here we have a highly speculative technology, which actually all our technologies are. And if we didn't get the investment to develop it, it wouldn't exist today. And basically, so it could be used in the developing world. In fact, we're doing some of that with the Gates Foundation. And anyhow, Alchemy's acquired, bought the company. And then there was a company called Civitas, which was spun out, and they were acquired by Accorda. The next slide just shows an example of a product that was approved. It's actually really pretty simple. Basically, you have this tiny little inhaler. This actually was approved by FDA, just as an example, to show you, for Parkinson's disease. And you can load up to 70 milligrams, by far the most that you could ever do for any inhaler before this, you know, based on this. And people are looking at it for other drugs too. On the next slide, I'm going to give you a third example. This is where we created a company to create new polysaccharides. And that's really complicated. You know, proteins, there plenty of companies have been based on proteins. They're called, proteomics type companies. Then a lot of you, I'm sure, have heard of genomics companies that is with DNA. But both proteins and nucleic acids are what we call monodispersed. In other words, if you had insulin, that's a protein. All insulins are about 6,000. They all have the same molecular weight. They all have the same structure. Polysaccharides, which is the third type of biomolecule, is much more complicated. It's actually polydispersed, and in any approved polysaccharide and any approved glycoprotein, what'll happen is, because of the sugars, it's polydispersed. It's actually a mixture. And it can be different molecular weights. A very good example is heparin, which is a polysaccharide widely used and by millions of people for anticoagulation. And there are many different molecules of different sizes and different amounts of the different sugars in them. At any rate, 2 of my students and postdocs, Ram Sasakaran and Ganesh Venakatraman, they were interested in taking the work that they had done and forming a company here. And what we had done, next slide, is we made some discoveries. We actually the team developed the first sequencing approach for any complex polysaccharide. This was published in Science. And the way it was done is using a combination of molecular scissors that would cut the sugars. The polysaccharides, you cut them in specific places with heparinases, heparanases, and things like that. And then what you do is you'd sequence the different cuts with mass spec, and then you put them together with a mathematical model that they developed in the Science paper. And that allowed you, for the first time, to sequence any polysaccharide or any really complex molecule. Now, what that means, next slide, is you might make new products like heparins, which was the molecule I mentioned, other complex polymers, or even new glycoproteins. And the next slide shows the company we started. It was with the two students that I mentioned. It got venture investments, and it went public in 2004. But the way these companies work is that you want to, again, as I said, it's a kind of a platform. You can do this with different polysaccharides. So they made deals with different larger companies to help fund them. And that helped fund a lot of these things. They got funding from Novartis, then Baxter, then Mylan, then CSL. And actually what was very exciting is that Lovenox, which is a low molecular weight heparin, that was the first biogeneric ever approved by FDA. And there were many, many much larger companies working on it. And yet Momenta, because we had the science and the patents, was way earlier getting approved than anybody else. So it was actually the first complex drug approved based on analytic data, and it was the largest syringe launch in history. Copaxone, which is another complex polymer, was also done by the same way, and that also got approved by FDA. The next slide just shows a picture of the enoxaparin that was made by Momenta, and what happened, as shown on the next slide, not unexpectedly, is that when they got this landmark FDA approval, the company did very well. I want to tell you one other story about Momenta, though. In 2008, there was a big crisis. Some heparin came in from China, and it was apparently didn't work very well, and it led to a lot of deaths. I'll just show you some headlines in the next slide. And it basically, people died in actually over 22 countries, or hundreds and hundreds of deaths around the world because of tainted heparin. Now, I had at one point been chair of the FDA's science board, their highest advisory board, and Ram had also done a lot of work with them. So at any rate, they asked me and him to see if we could help. And so we worked with Momenta, and actually Bob Linhart, who was another one of my former students, a professor at RPI, and we wrote 3 papers, and those are shown on the next slide. The bottom paper shows the contaminant. It was oversulfated chondroitin sulfate. That was published in Nature Biotechnology. And the other 2 papers, published in the New England Journal of Medicine, were published not only out of MIT, but also Momenta, the company, and also the CDC, the Center for Disease Control, and the FDA, Food and Drug Administration. And after we published all this, then there were no more deaths. So this was actually a really good example of where you could use science that was developed by our students and by the company to create to solve a major public health problem and really reverse it. So I was going to do one more medical example, and then I'm going to go over some other things. The next, if we go to the next slide. So Klaus Jensen, who was one of my colleagues at MIT, he was actually head of chemical engineering when we started this. We had written a grant to better understand how different forces could drive things into a cell. And you could look at things like electroporation or cell-penetrating peptides or nanoparticles or liposomes, but all of them had issues. And if we just press a button, you'll see what some of the issues are. If you can press one more button. And yeah, So now I want to show you what we did. This is a video, and I may actually have you play it back. But what we did is we forced cells through this little tube. Klaus does a lot of stuff on microfluidics. And what you're seeing on that picture, I'll just, we'll just look at it for a second, if we could stay there, please. So what you see is that there's this kind of tube, and then you see a constriction, and then you see like this, this injection. We're doing like a micro injection. So Armin Shari, he was our student doing his PhD thesis, and he was just studying the basic mechanisms. But one day he decided to take away the injection system where we're doing the micro injection. And when he did that, he got exactly the same result. Why don't we just show the cell going through the constriction just so people can see it? Yeah, you see the little circle going through the constriction. So basically what was exciting is he got the same result whether he did the injection or not. And that was fascinating. So what it told us is that deforming the cell, rapid mechanical deformation or squeezing it, must be responsible for allowing stuff to get in. So if we go to the next slide, what he did is, and we published this in Proceedings of the National Academy of Science, is he basically created a system where he could put 1 million cells per second through these chambers and deliver almost anything into the cell by squeezing it. And on the next slide, just as an example of this in the paper, he looked at, Dr. Yamanaka from Japan. He had won the Nobel Prize actually for showing that you could reprogram fibroblasts to make them look like stem cells by adding 4 genes. And you could check this by colonies. And basically they call what he did iPS cells. And basically what we looked at is, well, how does the different approaches work? Well, the cell penetrating peptide, that's CPP, that got 2 colonies. Nucleofection, that's the name for electroporation, get 11 colonies. And our device, Armin's device in the paper, got 150. So it was by far the best one. And it actually also turned out to be the safest, not only in terms of cell death, but on the next slide, you can do something called looking at the transcriptome. This was published in PNAS. And the transcriptome is the sort of the messenger RNA profile. And if you look at this gel, if you look at the control where nothing's happened, you see a pattern. It's like a fingerprint. But when you do electroporation, which is actually the gold standard has been the gold standard up till now, you see enormous changes. Whereas if you squeeze the cells, the changes are far, far less, and yet it's more effective. It turned out actually in 2014 that Scientific American, they have a vote, I guess, about what was the top 10 world-changing ideas of the year before. And on the next slide, they picked CRISPR-Cas9 as the number 1, and this squeeze is number 2. I always kind of make a joke that they got the order wrong, but it's okay. So this is how it looked, and we started this company, Squeeze. Actually, Armin, who was my graduate student, he became the CEO of it, and he is the CEO of it now. And but we got Amy Schulman, who is a really experienced businesswoman as executive chair, and she has provided terrific advice. And the company has done very well. It now has over 100 people. And actually, they've gotten a giant deal with Roche. This is shown on the next slide. And basically what they're able to do is make B cells. People have probably heard of CAR T cells. Well, this is a way that maybe you could make B cells, maybe T cells as well, by using this squeeze approach, which is, like I say, safer and more effective. So that's now in clinical trials for cancer, which is very, very exciting. Well, the last example that I want to give is very different. We've been fortunate to have a lot of venture capitalists help us and give us a lot of funding to get these companies off the ground and to get these products to patients. One day, one of those venture capitalists, by the way, I should say quite a few of those venture capitalists have been my own students. One of them is Amir Nishat, and he's now heads Polaris Ventures, which is one of the biggest venture groups in the country. And he came to see me one day with one of his partners. He said, well, Bob, we were interested in starting a hair care company. And I said, well, what kinds of things are you interested in? He said, well, one of the issues is that a lot of people get frizzy hair. If we could go to the next slide. So he said, could we come up with a way to prevent frizzy hair? And we started this company, Living Proof. If we could go to the next slide. So the way we approach this is the way we approached all the science that we do, which was we started to look at the fundamentals. And other people before us, of course, there are probably about 50 companies, next slide, that had created no anti-frizz products. And you can see them in various magazines or newspapers. Interestingly, they all do exactly the same thing. They use silicones as the main ingredient. They formulate them differently, but they that's what they do. They use silicones, and they're oily and greasy. They don't really control humidity, which is the problem. In fact, that gets to the mechanism. What causes frizz? Well, if we go to the next slide, what causes frizz is humidity. And if you put silicone on a hair, you'll see on the next slide that it doesn't really fight humidity very much if you look at an isotherm. So we took a much more fundamental look at this. We said, well, if you really want to prevent frizz from happening, you need something much more hydrophobic than silicone. And we thought the most hydrophobic thing we could think of was, well, it was actually like a Teflon frying pan. But of course, you certainly don't want to use that. But still, there's chemistry there that you could use. And we said, well, have any of these things been approved by FDA? And we found actually what's called a polyfluoroester, which is part of the family that the Teflon's in. And we took a polyfluoroester and we put it in and as an ingredient. Again, you don't need very much of it. And the next slide shows you that in contrast to the silicone, next slide, that makes a huge difference. So the company came out with a whole bunch of no frizz shampoos and conditioners. You'll see this on the next slide. They're sold at Sephora stores, and people really like them. And I thought I would just show you a video where from a TV show where they actually talked about this. If we could just turn on that video. Thank you.
Better to seek out today's leading edge of invention? It's a complete no-brainer. MIT.
We've done a lot of work on new kinds of implants to treat brain cancer, to treat different kinds of cancer and other diseases.
Bob Langer and Dan Anderson run one of the biggest academic research labs in the country. Langer alone has over 750 medical patents, more than anyone in the world. He and Anderson have had amazing success inventing novel medical materials and life-saving therapeutics.
Well, certainly it's one of the reasons I really like my job. I mean, you really feel like the work you're doing on a day-to-day is not only scientifically interesting, but also can have a real impact on people's lives.
Now, their work on stem cells and damaged spinal cords has had to make way for another vexing problem. Bad hair days. The result? No frizz. Based on Anderson and Langer's science, no frizz has taken the beauty world by storm.
It's so amazing that the big companies haven't been able to solve these problems in such a short time. We've been able to really tackle problems that have really dramatically affected women for many years.
Rob Robillard is CEO of Living Proof, makers of No Frizz. This Cambridge startup secret weapon, science. Using renowned researchers from outside the beauty industry to apply new thinking to age-old problems.
This side of her head was treated with one of those silicone products. Yay. This side was styled with no frizz technology.
Okay. We found the holy grail for frizz. It's amazing.
Noted stylist Mitch DeRosa is a co-founder of Living Proof. He says until now, the fight against frizz has relied on silicone-based products, which keep moisture out, but can leave hair heavy and greasy. Along come Anderson and Langer and their discovery of polyfluoroester.
This creates a weightless shield sheathed around each hair fiber.
For Langer and Anderson, there's nothing trivial about their work for a beauty product. After all, they consider themselves equal opportunity problem solvers.
Do your fellow professors teach you sometimes about the Frizzies, or they have a fun with it, or they?
A lot of them use it. So at any rate, so that's just an example. Also, another thing that they asked us to do, the venture capital people and the hair people, they said it would be really good if you could give hair more body. So Dan Anderson, who you saw was a postdoc with me, he's now a tenured professor, full professor at MIT. He and I and our students had designed these polymer libraries for different things like gene therapy and so forth. They're called polybeta amino esters, and we had a whole series of patents, like composition of matter patents on them. And they are actually used today, some of them, as reagents for gene therapy. But since we had them, we thought we could screen them for other things as well, and we did. And we found one actually gave hair a lot more body, and that you see in this with this woman, this blonde woman, and the product shown on the next slide. And so we actually had a whole line of products. I think Living Proof must have close to 100 products. Those are a couple of examples. But one of the key things I learned, for medicine, I think one of the things, what really matters is the FDA and how good your clinical data is. The marketing, however, really ends up being key. We hired a woman, Grace Ray. She later even she's a Harvard MBA. Later on, we hired her as vice president of marketing. Later on, she became president of the company. And I remember one time she, when she was talking to me, she said, something like 95% of the women who look at our products like them. She said 95% roughly reuse them. She said, but less than 1% of the people know these even exist. So what she and Jill, who was a CEO at the time, did is they decided we needed kind of like a spokesperson. So they got the spokesperson with really good hair, and she became involved in the company too. Here's a picture of her and me on the next slide. And if we could go to the next slide. So anyhow, because we had her, now we had both a good scientific product and somebody that was well recognized by the world, and its company did extremely well. Also, another thing we invented, if you look closely, we published this in Nature Materials, was we had this this woman, we came up with a cream. Betty Yu, another one of my students, did this with her team, and it was a cream that you could put on, and it could get rid of wrinkles. And basically, when you look at the right eye, you see the woman having wrinkles, but the left eye looked like that. But then when we put this cream on, which is another polymer system, it really goes away for something like a day. At any rate, here the investors, they sold the hair care company to Unilever, and for a very substantial multiple, and then they sold this part of it to Shiseido, a giant Japanese cosmetics company. So those are the examples I wanted to give you today. I thought I'd just summarize a little bit by giving you a list of some of the companies we've been involved in over time. I mentioned some of them. Enzotech was the first one, but we also developed food companies. We developed tissue engineering companies, all kinds of medical device companies. The next slide shows some other ones that we did over time. And again, some of these I've gone over, like Momenta and Living Proof, but we also created a PureTech, which has gotten FDA approval for new gels that can get rid of, anti-obesity and things like that. T2 Biosystems, which has FDA approved ultra rapid diagnostics for different bacterial panels. The next slide, and actually, I believe this is the last slide, shows, if we could get this, some of the other companies. In fact, just to give you a couple of things that you may have heard about, Taris was urologic delivery that got acquired by Johnson & Johnson. Kala has developed new nanoparticles that have led to new treatments for dry eye and other things. Moderna, a lot of people may know of, it's in the news a lot lately because they have the first, when we're talking about the coronavirus, Moderna developed the first vaccine that's now just started clinical trials for treating that, and so forth. So they go on and on with different companies that we've developed, and there, continue to be more. But basically, that's kind of what we've done, and it's a lot of it's really all of it started in our lab, pretty much, or at least a great deal, and it's led to a lot of things where our students have played a major role in terms of creating new companies and really new products that have affected the lives of many, many people around the world. So I want to thank you very much for having me. I wish you all the best, and thanks so much. Take care.